Right, aerobic respiration. These few slides here are literally only to help you keep the overview. It is very easy to get distracted by all those chemical keywords, the processes, where does this, where does that take place, that actually the overview and the purpose and the reason for aerobic respiration gets forgotten. So bear with me, this is going to be just the overview. We will have separate sessions on all the individual parts and individual, where does what chemical process take place. So for now, keep that overview thought in mind. So the aim of aerobic respiration, I'm pretty sure that at the back of your mind you still have, oh, that's the thing with glucose and oxygen coming in, and you get as waste products carbon dioxide and water, and it happens in the mitochondria, the powerhouse of the cell. If you write down powerhouse of the cell, that's for A-level standard, just 0 marks. So you'll need to step well beyond that. It has to be aerobic respiration for mitochondria. The aim of it is to release energy, and to release that energy in a format and in a package that the cells can actually use. I could give cells various forms of energy and see what they're doing with it. Let's see if we can give an animal cell some light, see what it does with it. Let's see if we can give a plant cell some petrol, see if it copes with it and gains anything from it. It needs to be the right format of energy that cells can actually use, and ATP is that format. It is a very small chemical. Think of it as a little bit of a rechargeable battery. It's a chemical that you can charge up again. It then releases the energy, let's say at an active transport pump, or it's really a small process that can be released, something that can be produced or synthesized somewhere in the cell, protein synthesis for the amino acid attachment, whatever small process that it is. The ATP is exactly that tiny energy package that a cell can use. Glucose is actually a large molecule, C6H12O6, that has a lot of energy that can be released when all those bonds are broken. So we'll need the energy released. Aim of aerobic respiration is energy in a form that the cell can actually use it. I called ATP a rechargeable battery. On a chemical level, you're attaching a third phosphate from an adenosine diphosphate to triphosphate. You're creating a high energy bond. The energy that's contained in that bond can be released around the cell as it changes again from ATP to ADP. Each glucose molecule in an ideal setting for aerobic respiration can release 38 ATP's worth of energy to recharge that. Anaerobic respiration releases only 2. Now that means aerobic respiration is definitely a better option if possible to release 38 ATP. So now come the steps. This is the overview of the steps that actually need to be taken so that a large molecule like glucose is broken down, energy packages pinched and pinched and pinched and pinched until we have got a system of energy release that we can actually get those ATPs. The overview, it's literally, ignore all the little niggly details in between. I need you to see the bigger picture. We're starting with a 6-carbon sugar. We are in the first step, which is called glycolysis, so breaking of glucose. We are turning a 6-carbon sugar into 2 3-carbon sugars. That's literally breaking up the sugar to make it a bit more accessible. Next step, the 3-carbon sugars are now actually the food for mitochondria. Mitochondria cannot take in a 6-carbon sugar. The 3-carbon sugar that you've made in glycolysis is taken in, and this next step is called the link reaction because it kind of links it outside to the inside and it links it to the Krebs cycle. We'll do that in a second. It's a change from a 3-carbon to 2-carbon compound. The niggly details in between, we will do them later. So the first step was your glycolysis. You break the 6-carbon into 2 3-carbons. As of here, literally, we do everything twice because we've got a 3-carbon sugar and another 3-carbon sugar. Each of them has been broken down into a 2-carbon sugar. The 2-carbon sugar also gets attached to a little enzyme, coenzyme A. It cannot otherwise be fed into a big chemical reaction, which is literally reaction after reaction, reaction after reaction, and eventually the whole thing becomes a circle because the starting point is kind of the end point again. This reaction here is going to be one that we spend a lot of time on. That's your Krebs cycle or TCA cycle or citric acid cycle, whichever way you want to call it. It's got various different names. Idea is your 2-carbon sugar combines with a 4 carbon sugar. 2 plus 4 gives you 6. Here we go. That's a 6 carbon sugar. And from here, it's a game of pinching and pinching and pinching and pinching. You're pinching away an energy package. You're pinching away an energy package. You're wasting little carbon dioxide. Oh, that's waste product. That's going to be just exhaled or diffuses outwards. You're pinching away energy packages. So it's literally all the way pinching away little energy packages. We will deal with what happens to those energy packages in the last step. But key idea here is you had your glucose, you broke it, and you broke it so that it is in a format that it can actually feed into this cycle. The 2 carbon can be fed into this cycle. From here, it's just energy pinching. Now, what do we do with those little energy packages? These energy packages, again, think of them as a different form of rechargeable battery. They're actually 2 different ones you need to be aware of, NAD and FAD. They pinch away the energy packages and take them somewhere, do something with it. Now what do they do? Again, this is a side note, we will do the detail on that. The energy packages, that NAD and that FAD, they're literally transporting energy in the form of an electron. So we're talking about the FAD becoming reduced and NAD becoming reduced because you're reducing the charge by attaching an electron, but that means you also attach the hydrogens to it. You need the enzyme dehydrogenase and so on. Leave that on the side for now. They are energy carriers. They're bringing the energy to specific pumps to power the pumps. That's their main purpose. So from the 3 steps so far, if you looked at the niggly detail in the very first step in the glycolysis, there's actually NAD being made. In the second step, in the link reaction, there's NAD being made as well. But in the Krebs cycle, in that 3rd step, lots and lots and lots of energy carriers are made. So what do these energy carriers now do? Right, these energy carriers are coming to a membrane. Now this is now you need to figure out where you are. We are in the mitochondria. We have been in the matrix of the mitochondria. The inner membrane and the outer membrane of the mitochondria separate out a space. So if you look at this one here, the inner and the outer membrane separate a space, the intermembrane space. Now this intermembrane space is the interesting part. We want to shove a whole lot of positively charged hydrogens, protons. That's literally a hydrogen, you've pinched the electron away. We're shoving a whole lot of protons into that intermembrane space. The aim of it, at this point you don't know it yet, this is now going to be the last step, that's kind of the important bit. You're using all those energy packages. If you look at the bottom here, you've got the NAD arriving and the FAD arriving at the pumps. The little green things here are the pumps in the membrane. That's the inner membrane of the mitochondria. I think that kind of sits on the cristae, and you've got the large surface area because they're all folded because we need to fit a lot of transport proteins onto that. The idea is that the energy carriers that arrive, the NADH and the FAD here that will arrive at the second pump, they will push the protons into the intermembrane space. The aim is to build up a gradient. So compartmentalized in the intermembrane space, you're going to have a build up of protons. So literally against where they want to be. You've got same charges next to each other. They want to repel. Diffusion wise, having the same chemical next to each other, that is again kind of diffusion. It wants to spread out. It wants to separate. So if you open up a channel, and allow that gradient to come out again, you can actually, like a bit of a wind turbine, attach something to it that uses that gradient. So we have a gradient. We have the intermembrane space of the mitochondria pumped full of protons, pumped full of those H pluses. Now they want to come out. This is something called chemiosmosis. It's a combination of electrostatics. 2 positive charges don't want to be near each other, especially 50 or 100 positive charges. They want to repel, they want to be away from each other. The other thing, diffusion. If there's a lot of one particular chemical in one place and it's able to move, then it would like to, if it could, move away from one another. Now we've got the phospholipid bilayer, so the proton cannot really get through that as it's got all those hydrophilic tails. Now if we allow one specific area where those protons can come out, it's a molecule called ATP synthetase. and it synthesizes ATP. It makes ATP. It uses that gradient from the protons coming back outwards, and the idea of a wind turbine, that's maybe not a bad analogy at all. It is a molecule that has been found to actually twist as and when those positive charges come through. You have done a little bit of protein structure, and you know that different charges can change the shape of a protein. In this case, it changes the shape in a way that it actually causes rotation. That rotation on the far end causes a pushing two chemicals together, pushing the ADP, the adenosine diphosphate with the two phosphate groups, and another phosphate into each other, because actually they don't want to be near each other. You've got the already two positive phosphates on the ADP, and you want to push a third positive phosphate onto that. That's not a bond that they want to make, so that it requires a bit of energy to overcome that electrostatic repulsion there, which means you're making a high energy bond, and that's exactly what you want ATP to have. You want it to have that high energy bond. That is the recharging station here. That's where ATP gets recharged. So by all means, use different resources. This is not a new A-level topic. I just want you to keep the overview over the whole thing. You've had glucose. You want a very, very small energy package. ATP is a small energy package that cells can access. Glucose is too big. So what do you want to do? You want to break your glucose. Break it from a 6-carbon into a 3-carbon. Break it from a 3-carbon into a 2-carbon. Obviously, at that point, you've moved from the cytoplasm into the matrix of the mitochondria. The 2-carbon then gets attached to an enzyme which just brings it into the circular reaction, that Krebs cycle reaction. Along the way, and every step along the way, small energy packages get pinched and pinched and pinched in the form of FAD being reduced and NAD being reduced. We will look at the details. So those energy packages, small energy packages, again, think of them as a different format of rechargeable battery. They are then going to the inner membrane. Along the inner membrane, you have got the pumps that are pumping the protons into that intermembrane, where the proton gradient will build up. The protons then flow back out. If you look here in this diagram, we've got a little black ATP synthetase. That's where the protons will flow back in, and while they do so, that ATP synthetase creates the ATP. Keep the overview. We will do all the niggly bits as we go through the next couple of lessons.